And get started. So my name is Tori Poplaski. I am currently the Vice President of ASM at the TMC, and I serve as co-chair of MicroTalks with Lynn. So just to quickly introduce MicroTalks for everyone, it is a monthly seminar series hosted by ASM, and we usually do it every third Monday of the month at the same exact time. And it's a really good platform for students and postdocs, as well as faculty, especially new faculty, to get to share their research with the wider community throughout the TMC. And ideally, we would like it to be unpublished data, and it's a really good chance for people to get feedback on different aspects of their research and increase collaborations throughout the TMC. Also, if you would like to sign up for a MicroTalks, Lynn posted the link in the chat. So please sign up for that if you want to give it one in the future. And with that, I'll turn it over to Lynn, who's going to be the moderator for today.
All right, thank you, Tori. So welcome to July's MicroTalks. My name is Lynn Su, and I'm the lead coordinator of MicroTalks, and I'll also be your moderator today. So bacterial pathogens have evolved a wide variety of tools to establish themselves in the host and gain nutrients, which also cause damage and disease. Therefore, it is very important to elucidate the different mechanisms that pathogens undertake during infections. So today we will have two talks that cover the microbial pathogenesis of B. anthracis and C. difficile. First, we will have Ileana Corsi, who will be speaking to us about small RNAs of Bacillus, anthracis, Virulence, Plasmid, PX01, regulate gene expression in trans. Ileana is a Ph.D. candidate in the lab of Dr. Theresa Koehler at McGovern Medical School. And following that, Dr. Jahangir Alam will be speaking to us about C. difficile infections, or CDI, the sources of pathogen around us. Dr. Alam is an assistant professor at the College of Pharmacy at the University of Houston. Okay, so with that, let's welcome Ileana.
Okay, thank you, Lynn. So I'm gonna share my screen real quick. All right, and then I'm gonna minimize you guys. All right, is everybody able to see that, or is Lynn, are you able to see it?
Yeah.
Okay, awesome, thank you. All right, so hi everyone. Thank you for coming to hear my talk. So I'm gonna tell you about small RNAs of Bacillus anthracis and my work in the Koehler lab. So small RNAs are versatile regulators of gene expression in bacteria. They typically function at the post-transcriptional level to regulate gene expression by interacting with mRNA transcripts. And those interactions will result in either negative or positive regulation of gene expression. So for example, in terms of negative regulation, an siRNA can base pair with an mRNA transcript at or near the ribosomal binding site, or RBS, of the RNA, and thus prevent the recruitment of the 30th subunit of the ribosome and inhibit translation, resulting in negative regulation. siRNAs can also influence aspects of mRNA decay. So for example, an interaction with the siRNA through base pairing can help facilitate recruitment of a particular RNase, aiding in degradation of the transcript, and in some examples can even enhance the activity of the RNase towards the transcript, thus again resulting in negative regulation, this time by enhanced degradation of the transcript. In terms of positive regulation, and going back to the translation example, if, for example, an mRNA has its RBS occluded by secondary structure, base pairing with the siRNA, typically at the 5' UTR of the transcript, can help kind of reveal that structure and allow the RBS to become available for interaction with the 30th subunit of the ribosome and allow translation to take place. And again, going back to the RNA decay example, siRNAs can help to stabilize mRNA transcripts, for example, by base pairing in such a way that blocks sequences that the RNAs would otherwise recognize. And so these are really just a few examples of mechanisms of siRNA function, but they typically do perform these functions at the post-transcriptional level. Okay, so in our lab, we study Bacillus anthracis, which is a gram-positive bacterium and a mammalian pathogen, which causes anthrax disease. Its genome is comprised of a circular chromosome as well as 2 virulence plasmids, one of which is called PX01, which is kind of the focus of the talk for today. So within PX01, there's a pathogenicity island. And within this island, we can find the 3 anthrax toxin genes, which are shown here in green, which together form the anthrax toxin, which, as the name implies, is an important virulence factor of Bacillus anthracis. And within this pathogenicity island, we can also find a gene called ATXA, which encodes an important protein of the same name, which is the master virulence regulator of Bacillus anthracis. We know from extensive work in our lab that ATXA positively regulates a variety of virulence factors, including the 3 anthrax toxin genes. It also kind of serves as a pleiotropic regulator by repressing expression of many genes, including many that encode proteins involved in different aspects of metabolism, such as amino acid metabolism. So our group, as well as others, noticed that within PX01, either within the pathogenicity island or in proximity to that island, there were 2 transcripts that were highly expressed. They were unannotated, so there were no proteins associated with these loci. And these were kind of short transcripts of very short lengths. And so we hypothesized that these could potentially be small regulatory RNAs, and we designated them PX01 encoded regulatory RNA, A and B, so SIR-A and SIR-B, as I'll be referring to them for the rest of my talk. And we know from data from our lab that ATXA positively regulates both SIR-A and SIR-B. So this is a read map of RNA-seq data showing reads aligned to the entire length of PX01. And so I'm showing you gene expression in a parent strain, as well as the strain that has been deleted for ATXA. And so in the ATXA deletion strain, we see that we lose expression of the anthrax toxin genes, such as these 2 right here, but we also lose expression of these 2 predicted sRNAs that are highlighted with these red arrows. And importantly, we can restore expression of the sRNAs if we then complement back ATXA exogenously into this ATXA null mutant. And so given that ATXA positively regulates these sRNAs, we asked the simple question, do SIR-A and SIR-B affect gene expression in Bacillus anthracis? So one of the first things I did was verify that expression of SIR-A and SIR-B is in fact dependent on ATXA using northern blotting as a secondary method. So I'm showing you here northern blots probing for SIR-A and SIR-B in a parent strain of Bacillus anthracis, as well as the ATXA null. And so you can see right away that you have expression of both sRNAs in the parent strain, and you completely lose detectable levels of sRNA in the ATXA null. And I'm using 5S as a 5S ribosomal RNA as a load control. I also did race analysis, mapping the precise 5' and 3' ends of the sRNAs to gain some more information about their lengths and their sequence. And I found that SIR-A is 182 nucleotides long, while SIR-B is 224 nucleotides. And sRNAs typically range in size from 50 nucleotides to 500 nucleotides or even more. So these lengths are well within those ranges of what is typically accepted to be an sRNA. We also know that SIR-A and SIR-B are highly stable, which is not necessarily surprising perhaps for sRNAs. So if we treat growing cultures of Bacillus anthracis with rifampicin, which is an antibiotic that stops transcription, and then take samples for RNA extraction at these indicated time points post-rifampicin addition, we can track the decay of the transcript over time using, again, northern blotting. So I did this for SIR-A and SIR-B 3 times, and I found that both sRNAs have a half-life of 20 minutes, which is considerably long for RNA, indicating that they're pretty stable. And perhaps one of the reasons why they're so stable might have something to do with the fact that they're resistant to degradation by a particular exonuclease that is 5' monophosphate dependent. So in bacteria, primary transcripts have a 5' triphosphate, while processed or secondary transcripts have a 5' monophosphate. And an enzyme such as TEX or terminator exonuclease that is 5' monophosphate dependent can only degrade secondary transcripts, but it will not degrade primary transcripts. And so if we treat SIR-A and SIR-B with the TEX enzyme, we see that there is no decrease in SIR-A and SIR-B relative signal, indicating that they're potentially resistant to TEX. However, if we first treat with an enzyme called 5' monophosphatase, which will remove the 5' triphosphate and leave a 5' monophosphate, and then treat with TEX, we now see a decrease, a significant decrease in SIR-A and SIR-B signal compared to the untreated control, suggesting that the resistance to TEX was mediated by a 5' triphosphate. So in order to assess the regulatory function of these highly stable primary transcripts, we performed an RNA-seq experiment looking at the transcriptome of sRNA null mutants compared to the parent strain. And we can summarize these data as a volcano plot, where the x-axis is a log2 transformation of the full change in transcript expression between the two strains, and the y-axis is the p-value. And each of these diamonds represents a transcript. So we're looking at the entire transcriptome, and I've highlighted things that were differentially, significantly differentially regulated in black versus those that were not in gray. So comparing the transcriptome of a parent versus the SIR-A null, we found that there were 12 transcripts that were downregulated in the mutant and 38 transcripts that were upregulated in the mutant, suggesting that SIR-A primarily functions to repress gene expression. And we got a number of interesting hits that I could talk about all day. But for the interest of time, for the rest of my talk in a few slides, I'm going to focus on this particular target of SIR-A called INHA1, which is a secreted protease of Bacillus anthracis. As you can see, this particular target was highly upregulated in the SIR-A null mutant, again, suggesting that SIR-A represses expression of INHA1. Of course, I also looked at comparing the transcriptome of a SIR-B null to that of the parent strain. And interestingly, SIR-B had a minor effect on the transcriptome of Bacillus anthracis, where SIR-B alone appeared to only regulate expression of one target, which was a putative transcriptional regulator. However, SIR-A and SIR-B together appear to co-regulate at least some transcripts, primarily given by the fact that deletion of both sRNAs resulted in even more transcripts being differentially regulated. So in this strain, we had 19 transcripts that were downregulated in the mutant and 97 transcripts that were upregulated, again, suggesting that the sRNAs primarily function to repress gene expression. And I got some of the same hits that I got before for the SIR-A null, but there were also other transcripts that appear to only be regulated when both sRNAs are deleted. And so, for example, here, this other putative transcriptional regulator that also showed up, GBA3479, was very highly upregulated only in the double null mutant. But for the rest of my talk, like I said, I want to focus on SIR-A control of INHA1. So, like I mentioned, INHA1 is a secreted protease of Bacillus anthracis that has been extensively studied by our group as well as others. We know from data in from our lab that INHA1 modulates the Bacillus anthracis secretome by cleaving the anthrax toxin proteins as well as other secreted proteases and kind of modulating the protein levels, the levels of proteins in the supernatant fraction of the culture. We also know through work with our collaborators that INHA1 degrades host proteins in addition to Bacillus anthracis proteins. In particular, it degrades serum albumin and hemoglobin. And we believe that this is the degradation of these proteins serves as an amino acid source for cells that grow and a medium that has a chemical composition similar to that of blood serum. So again, suggesting that these proteins can serve as an amino acid source to support growth of the bacterium. Other groups have shown that INHA1 can help regulate attachment to endothelial cells, primarily by cleaving another Bacillus anthracis protein called BSLA. And other groups have shown that INHA1 may play a role in pathogenicity primarily due to NHA1 mutants being attenuated in a mouse model, as well as being impaired in brain colonization, suggesting that this protein has some role in modulating permeability of the blood-brain barrier. So like I mentioned, through my RNA-seq experiment, I found that SIR-A potentially represses expression of NHA1. And through in silico modeling, I found that SIR-A is actually predicted to base pair with NHA1 at the 5' UTR of the transcript. So if this is the NHA1 transcript sequence, and this is the start codon, or the translational start signal, and this is the 5' UTR, we see that there's this region of about 12 nucleotides that is predicted to base pair. And so this kind of interaction is kind of reminiscent of that first mechanism that I mentioned at the beginning of my talk, where interaction between the sRNA and the RBS of a transcript can block recruitment of the 30S subunit of the ribosome and thus inhibit translation. So I asked the question, does SIR-A inhibit translation of NHA1 by direct base pairing? And so one of the first things I did was look at this region of SIR-A more carefully to see whether it would in fact be available for base pairing. So if you take the sequence of SIR-A and put it through some kind of structure modeling prediction software such as mFold, you can obtain a predictive structure that looks kind of like this, showing a high degree of base pairing and secondary structure. And in fact, this region of SIR-A that is predicted to base pair with NHA1 is found right at the 3' end of the transcript, and according to this prediction model, appears to be double-stranded. So for that reason, we decided to look at the structure a little more carefully and do shape analysis of SIR-A, which stands for selective 2' hydroxyl acylation analyzed by primary extension. So we take in vitro transcribed RNA and treat it with benzoyl cyanide, which will mediate 2' hydroxylation of nucleotides on the RNA. And this modification will only occur in nucleotides that are single-stranded. And then we can map these modifications using primary extension. And so when you do this, you get a shape reactivity score for each nucleotide of the entire RNA, where the higher the score, shown in red, the higher the probability that this particular nucleotide is single-stranded. And then you can take all that data and fit it through a folding prediction model again, and then obtain kind of an experimentally informed structure instead of just a prediction. And so looking more carefully here at this region that is predicted to interact with NHA1, we see that it is in fact predicted to be single-stranded. And so it's most likely available for interaction and base pairing. So I went ahead and created a translational fusion of the 5' UTR of NHA1 to GFP under the control of a constitutive promoter. And this, of course, includes this region that is predicted to base pair with SIR-A. And then looked at GFP expression in a parent strain of Bacillus anthracis and the SIR-A null. And I found that while in the parent strain I get some level of GFP production, in the SIR-A null I get this increased level, which suggests the repression of this region. And I also did a control experiment just making a translational fusion of GyrB to GFP, which is an enzyme that is, according to my RNA-seq data, not a target of SIR-A. And I, of course, see no difference in GFP production. And so finally, we were interested in the potential effect of these sRNAs in virulence of Bacillus anthracis, given that they appear to regulate many targets, including apparent virulence factors of Bacillus anthracis. So we infected AJ mice, which are C5 deficient, with 10 to the 5 CFU of Bacillus anthracis of either the parent strain, the SIR-A null, or the double null, and then tracked survival of the mice over time, and then also extracted organs at the time of death to look at colonization of organ tissues. We found, however, that infection with the parent or the sRNA null had no effect on survival of the mice in this systemic model. However, looking at organ colonization, we found that in the lung, the liver, and the brain of mice, there was a slight defect in colonization in the sRNA mutant strains. And so suggesting that these sRNAs potentially are modulating some aspect of survival in the host. And we're going to explore this further with more, with further experiments. And so in conclusion, today, I've told you about SIR-A and SIR-B, which are the first sRNAs to be described in Bacillus anthracis. I've told you that these sRNAs are under the control of ATXA, which is a master virulence regulator of Bacillus anthracis. These are encoded on a plasmid and appear to regulate many transcripts that are encoded on the chromosome of Bacillus anthracis. SIR-A appears to have distinct roles regulating many transcripts, while SIR-A and SIR-B together appear to have some overlapping roles. And I've also shown you that SIR-A appears to directly base pair with NHA1 to regulate the protein levels. And this is the first evidence that we've seen of sRNA-mediated control of NHA1. And so we still have many, of course, remaining questions. And some of those questions that I plan to address in the future are looking at what other transcripts could SIR-A and possibly SIR-B base pair with, and could these sRNAs also interact with regulatory proteins. And so with that, I'd like to thank my lab, Terry, my boss, and members of the Koehler lab, both past and present, as well as Ed Economics from Rice University for helping us with the shape analysis, Amber Van Hoof from my department for helping me or teaching me how to do RNA-seq analysis, and Nick for his help with race analysis as well, and teaching me how to do that. And with that, I'll like to take any questions.
Okay, thank you, Ileana. So just to remind everyone, if you have any questions, you can post them in the chat. Okay, so we have one question. What is the most exciting part of this research to you?
The most exciting part? I think the most exciting part is that we don't really know anything about small RNA regulation in Bacillus anthracis. You know, we've studied anthracis for many years. Our lab has studied anthracis for many years and studied toxins and different virulence factors. But this is actually the first time that we've seen small regulatory RNAs influencing those virulence processes in some way. So it's kind of new, and it's kind of new experiments that other people in the lab haven't necessarily done because it's focused on RNA versus protein work. So it's been exciting to kind of validate protocols and create new protocols for the lab to do these kinds of experiments.
Great. I have two questions.
Mm-hmm.
So from the volcano graph, is that the right term?
Yeah.
Like, so are the SIR-A targets from RNA-seq, are they all related in functions, or are they kind of like, because you only talked about the top one, right?
Yeah, sure. So some of them appear to be related. So NHA1, like I mentioned, is a protease. There were a few other proteases that also showed up in this analysis. There were also different aspects of branching amino acid biosynthesis and transport that were affected. And so those are related. There were also different oligopeptides that appear to be also repressed by SIR-A. So in general, it does seem like SIR-A in some way regulates amino acid metabolism in a way. Mm-hmm. So proteolysis and then uptake of those peptides that are produced from proteolysis, as well as synthesis of amino acids as well.
Mm-hmm. Okay. And then another question that I have is just, so did you, because I know that you checked just SIR-A with the INHA1 protease, but what about like SIR-B alone, like SIR-B knockout?
Yeah.
The target?
So SIR-B alone didn't affect NHA1. It really only appeared to affect one transcript. And so really by itself, it doesn't really seem to be doing much. But when we delete both SIR-A and SIR-B, there are a handful of transcripts that appear to be regulated only when we delete both. So
Okay.
there are some things that are potentially regulated by both siRNAs together.
Okay, got you. So, so does anyone have more questions? All right. Okay. Thank you, Ileana.
No problem.
All right. So let's welcome Dr. Alam for the second part of the talk.
Thank you. So today I'll talk about about the sources and reservoirs of an important diarrhea-causing pathogen, Clostridium difficile. It's mostly environmental studies, so I want to start from here. So as you know, I'm an environmental microbiologist working at the Department of Pharmacy Practice and Translation Research. And we have a infectious disease group in that department at College of Pharmacy at University of Houston. Dr. Kevin Gary is the team leader of this group, and we do many different kinds of research together. So mostly we do research on Clostridium difficile, infection research, clinical and environmental aspects of Clostridium difficile, from molecular to environmental. We try to do everything about C. difficile. Other than that, we have research on vancomycin-resistant enterococcus, carbapenem-resistant enterobacteria, CRE, MRSA, Candida, and other pathogen research. But today's talk will mostly focus on environmental aspects of Clostridium difficile. The objectives of our environmental studies are to understand the ecology and epidemiology of pathogenic C. difficile in One Health approach. We try to determine the prevalence and characterize the pathogen C. difficile from many different sources. Today, I'll mostly try to correlate the prevalence of this pathogen in the environment, and that environmental presence of this pathogen is mostly associated with fecal contamination, animal or human fecal contamination. So what is C. diff? Clostridium difficile. Clostridium difficile is a gram-positive, strictly anaerobic, endospore-forming toxigenic bacteria. This is a serious diarrhea-causing pathogen for human and some other animals. There is a new name for this pathogen that is called Clostridium difficile. As this is a spore former bacteria, these spores of this pathogen can survive anywhere for many months. Therefore, these spores can be source of infection or colonization, colonization of human and animals. They mostly colonize the gut, and they can also grow in the intestine of many animals, and these bacteria can disseminate in the environment through their feces. So we regularly culture this strictly anaerobic bacterium in our lab from many different sources, from CDI patients, Clostridium difficile infected patient stools, from animal stools, from environmental swabs, from everywhere. We try to understand the ecology and epidemiology of this important pathogen. Therefore, we try to sample all kinds of sources. And for that purpose, we need a really good anaerobic system to culture them, because this bacterium cannot tolerate oxygen and they will not grow in presence of oxygen. And we also need highly selective media to find them from thousands of other bacterial backgrounds. And this bacterium has two groups, toxigenic and non-toxigenic. Toxigenic strains can cause disease, but non-toxigenic strains cannot cause disease. There are several toxins they can produce, enterotoxin, cytotoxin, and another is binary toxin. Binary toxin produced by a specific group of strains, but most of these strains actually produce cytotoxin and enterotoxin. There is another interesting fact about this bacterium, Clostridium difficile. A group of scientists from Germany and New Zealand, they discovered that this Clostridium difficile is an autotrophic bacterial pathogen. It means they can grow on carbon dioxide and hydrogen as sole carbon source, carbon and energy source, thus representing the first identified autotrophic bacterial pathogen. Mostly all bacteria are heterotrophic, but this is autotrophic exceptional bacteria. So maybe there are other bacteria we don't know. With time, the scientists will discover the other autotrophic bacteria. But interestingly, this is the first human pathogen, and it can also live autotrophic life. According to CDC, this pathogen, Clostridium difficile, is an urgent threat level pathogens. These are high consequence antibiotic resistant threats because of significant risks. These threats may not be currently widespread, but have the potential to become so and require urgent public health attention to identify infections and limit transmission. How common are these infections? Clostridium difficile is a major health threat. In 2017, there were estimated 223,900 cases in hospitalized patients and 12,800 deaths in the United States, according to CDC. This is just a rough estimate, but there may have many other infections in the community. Usually, most of the cases are in the hospital, therefore we call it hospital-acquired infections or antibiotic-associated infections, because when we take antibiotics, we disrupt our gut microbiota, and this bacterium can colonize us, they can grow, produce toxin, and these toxins are associated with severe diarrhea, colitis, and toxic megacolon. So this is really a serious pathogen. There may have millions of cases of this pathogen all over the world. There is no no country without this problem, but most of the people don't know about this pathogen. And in this country, this is a billions of dollars problem every year. And in this world, maybe it's much bigger problem. Another interesting fact is that recently we have noticed that there are COVID-19 and CDI co-infections in the hospitals. So it seems this coronavirus is messing up with our CDI patients. That is another interesting thing, because COVID-19 have diarrheal symptoms. Some patients may have diarrhea with COVID infection, and CDI also causing diarrhea, and therefore maybe it is messing up in the for diagnosis and treatment. Now I want to introduce our first study on environmental aspects of Clostridium difficile. That study published in 2014, and that was the first environmental study we started in Houston with our group. So in this study, the objective was to find the environmental contamination level in our house environment. And therefore, we collected some environmental samples like shoe bottom swabs, floor swabs, restroom, toilet swabs, kitchen counter, etc. Or even we also collected household dust to see the prevalence of toxigenic C difficile in our house environment. And we found high environmental contamination of C difficile in this study. Let me show you a little bit of that data. So for this study, we recruited a student, high school student. Here is she. Her name is Ananna. Actually, she is my daughter at that time. I recruited her to collect household samples from her friends' houses, and I trained her to analyze all these samples to find Clostridium difficile. And with this small summer project, the high school student project, what we have found, here is the summary of this data. Prevalence of, in this figure, I am showing the prevalence of C difficile from various environmental samples from our household. So here you can see 40% of the shoe bottom swab samples positive for toxigenic C difficile. It was surprising to us because it is grossly contaminated soaps. And then restroom or bathroom samples, 33%. Other surfaces, floor and kitchen surfaces, 13%. Even household dust contained 33% of toxigenic C difficile strains. Overall, there are 32% samples positive from this group of samples. After seeing these exciting data, we started another project on community environmental contamination of toxigenic C difficile in Houston area. This is our second study published in 2017. And for this study, the objectives of this study were to assess community environmental contamination of toxigenic C difficile and to assess strain distribution in environmental versus clinical isolates. For this study, we collected over 2,500 environmental samples over the study period. And here also, we identified a high prevalence of toxigenic C difficile from community environs that were similar ribotypes to clinical isolates. This finding suggests that intervention beyond isolation of symptomatic patients should be targeted for prevention of C. difficile infection. Here are some data from this study. Here you can see from park samples, from various parks in Houston area, we collected 491 samples, and 24.6% samples were positive for toxigenic C. difficile, like our previous household environmental samples. Again, we collected many samples from various homes, over 1,000 samples, and found 17% samples positive for toxigenic C. difficile. From chain store, 7.6%, fast food restaurant, 6.5%, and other commercial stores, 8.1%. So it seems C. diff are everywhere. These bacterial spores are sitting everywhere. And here is another figure showing the distribution of C. difficile in household environment. Again, shoe sole, 26.4%, doorstep, 24.7%, cleaning supplies, 13%, kitchen and restroom, 9%. So overall, again, we have found C. difficile spores everywhere in the house environment. So our house environment is also a good source of C. difficile pathogen, and we can get infection from these sources, or we can be colonized and later get infection from these household environments or park environments, or even from commercial stores. Here I should mention that these are grossly contaminated environment, I should say, because if we compare it with our normal human healthy people's stool sample, we can find only less than 5% samples positive for Clostridium difficile. So compared to human stool, normal human stool, the contamination level of these environments are much, much higher. Therefore, I should say everything grossly contaminated with fecal bacteria and Clostridium difficile. Clostridium difficile is a fecal bacteria. Most of the animals actually can be transiently colonized, and they excrete this pathogen with their feces. Now I should show some other molecular characteristics of this pathogen to understand the diversity of these strains from many different sources, like community environment, hospital environment, and clinical patient isolates. Here, you can see the proportion of various ribotypes of this pathogen is very similar in all these sources: community, environmental, hospital environmental, and clinical isolates. It means they are connected to each other. Means the patient grossly contaminate the hospital environment, and they may get the infection from the hospital environment frequently. And we also know that CDI infection is very prevalent in the community also. Therefore, the community environments may be associated with these community infections. Now I want to show some data from other studies done by European scientists. Here in this table, we can see the overview of recent European studies on C. difficile in animals. As animals are the main reservoirs of this pathogen, we wanted to see what are the ribotypes or strains circulating in animal population. Therefore, I am showing this data. Here you can see they studied pigs, cattle, goat and sheep, poultry, horse, cats, dogs, many other animals. And in the right column, you can see the reported prevalence and the most prevalent ribotypes. Here we can see the prevalence can be from 0% to 100%. So it's widely variable. Some animals can be highly colonized, and some may be free from C. diff. Overall, it depends on the age of the animals and their feed and health condition, and also may be associated with antibiotic use, because we use a lot of antibiotics in the agricultural system. So that may be enhancing the colonization of this pathogen in animal guts. Here is another table showing the European studies on C. difficile in foods, raw foods basically, means meats, seafood, vegetables, and in the right side column, you can see the prevalence and ribotypes. Here again, it's wide range of contamination in this meat, seafood, and vegetable products. And we can find from 0% to up to 75%, and all kinds of ribotypes are there. These are also clinically common ribotypes in human. And these are also available in various food items. So this can be a big source of infection. We also did another study on domestic dogs and zoo animals in Houston area. In this study, we wanted to see whether these zoo animals and domestic dogs can be source of Clostridium difficile, and what's the impact of antibiotic use in zoo animals. Here, you can see in this figure, we are showing the prevalence of toxigenic C. difficile ribotypes in zoo animals at baseline, during antibiotic therapy for some diseases, and at follow-up. So Clostridium difficile colonization increased significantly during the antibiotic time period. Here you can see the various ribotypes, and at the baseline, there are 14% samples positive for Clostridium difficile. And during antibiotic treatment, the colonization increased up to 33%. And then after 2 months of antibiotic treatment, that prevalence went down to 11%. So in animals also, antibiotic use cause C. difficile colonization in animal gut. Here is another table on prevalence of toxigenic C. difficile ribotypes in dog stool, dog paws, and community environment, means dog parks. So here you can see, although the sample number is very limited, we have found dog paws were positive for 56% of the samples. And in dog stool, it was 17%. And in park environment, dog park environment, community environment, we got 22% positive for toxigenic C. difficile. And we also found some important ribotypes associated with these samples. Then we wanted to see whether the birds are sources for Clostridium difficile, toxigenic C. difficile. You know, birds are everywhere. They can go in many places. When they sit on the trees, they poop. And here you can see tree leaves with bird poops. It's a common thing. And these bird poops dry up, and with the air, these poop can go anywhere. Therefore, our total environment is grossly contaminated from many different animals, including birds. So these bird poops can be a good source of infectious C. difficile. Therefore, we collected some tree leaves to see whether these bird poops are good source of C. difficile. Here, you can see the results. From the tree leaves with bird poop, we found 33% samples positive for C. difficile. Out of them, around 27% were mostly toxigenic, 27% samples with toxigenic C. difficile. It means these bird poops are loaded with toxigenic C. difficile. It's a big source of this pathogen. And then we wanted to see the ribotypes. What are the diversity of these strains? And we found important ribotypes associated with clinical isolates. And therefore, we believe these are associated with human infection also. And then we also wanted to see whether the soil samples, park soil samples or our backyard stool soil samples are contaminated with C. difficile, because we believe everything contaminated with animal feces, including birds, insect, everything. And we collected some samples and found that there are two types of soil samples, topsoil and the subsoil. On the topsoil, as it is mostly contaminated with fecal material, we found more toxigenic difficile from topsoils. Around 30% of the topsoils are contaminated with C. difficile spore. And then in the bottom soil, we got 14% samples positive for toxigenic C. difficile. It means soils are also grossly contaminated with C. difficile spore. So we can get infection from, or colonization, we can be colonized from soil sources also. And then we wanted to see again the diversity of these strains, and we found all important ribotypes in soil sample and subsoil sample. Therefore, I believe these are the good source of C. difficile infection. When we are weak, when we are taking antibiotic, we can get this infection or colonization of C. diff from many different sources. Here is another preliminary study of horse farm and house flies. In any animal farms, there are millions and millions of house flies, and these house flies can carry many different kinds of germs from animal feces. House flies can fly up to 30 miles from their birthplace, so they can carry any germs to other animals or food or in water or in vegetables, they are dangerous vector, really nasty vector of many different germs. Therefore, we investigated the animal stool for C. difficile and houseflies. And what we have seen that the horse fecal material is contaminated, is a good source of C. difficile. Here we got about 10% of the samples positive for Clostridium difficile in horse fecal material. And from the same field farm, horse farm, we collected 247 houseflies. And from these houseflies, we got 15 toxigenic C. difficile. So they are also colonized or carrying this C. difficile from animal feces in many different places. So this can be a good vector for transmitting C. difficile spores from place to place, from animals to animals, or animals to food. And here another study with shoe bottoms from another country, Malaysia, Sabah, Malaysia. And we collected 200 shoe bottom samples to see whether these are similarly contaminated like Houston area shoe bottom samples. And we found similar data. 16.5% of the samples are positive for toxigenic C. difficile. And the diversity of these strains are huge. Therefore, and they are also, these ribotypes also associated with human infection. Therefore, we believe, anywhere in this world, C. difficile is there. There is no place without C. difficile spore. And now I should say it's this fecal contamination. Animal fecal contamination is really important. Fecal contamination matter. And we can say C. difficile spores are everywhere, mostly coming from fecal sources. Human and animal guts can be transiently colonized by C. difficile. C. difficile strains are genetically highly diverse, and many of these strains are pathogenic. Therefore, we need to maintain good hygiene to prevent C. difficile colonization or infection. And I believe One Health approach is the best approach to study any infectious disease pathogen to understand the ecology and epidemiology of this of the pathogen A special note, I think everybody knows it, that is, we regularly use alcohol-based sanitizer to sanitize our hands. And these alcohol-based sanitizers cannot kill the spores of C. difficile. Therefore, we should not use alcohol-based sanitizer to disinfect our hands or any surfaces. We should use soap and water to clean our hands. That is really important. Nowadays, alcohol-based sanitizers are very popular in the hospital and in healthcare system, but that will not prevent or kill the C. difficile spore. And here are our future plan. Basically, we want to do everything from molecular to environmental aspects of C. difficile. So hopefully we'll be able to do a lot of other things on this pathogen. Here is a picture of our infectious disease research team. Dr. Kevin Giri is the team leader, and we do many different research together and publish together. And that's all for today. Thank you so much for listening.
All right, thank you, Dr. Alam, for the talk. So we have 2 questions from the chat. First question is, what controls do you do to make sure that the samples are contaminated with C. difficile in the lab?
Okay, we have controls. When we swab, we use that cotton gauze swab and the container and PBS, everything. We always have a subset as a control. During processing, during sample collection, for every step, we have such control to see whether we are contaminating, cross-contaminating the samples. So we are always doing it to prevent the cross-contamination or lab contamination for our samples. We are really good at to control all this cross-contamination.
And the second question is, what does One Health mean?
One Health means, these sources of pathogens can be very diverse. This pathogen, Clostridium difficile, can come from animals, from house environment, from air, from stool, from food. So if we only consider the patient, then we'll not be able to understand the ecology and epidemiology of this pathogen. We need to study everything. That is the One Health approach to control and prevent diseases.
Okay. And I actually have 2 questions myself. So, so what is the mutation rate for the C. difficile strains or the ribo types?
Overall, there are many ribotypes, already over 600 ribotypes reported by this time.
Mm-hmm.
There are many others maybe, but overall, this bacterium is mutating regularly.
Mm-hmm.
It's very diverse group of pathogen, but all kinds of strains can cause disease if they have toxin producing capability. Means if they have the toxigenic genes, toxin A, toxin B, and binary toxin genes, they are capable of producing toxin and cause disease. But there are some non-toxigenic strains, they cannot cause disease. Therefore, we are not afraid of non-toxigenic strains.
Okay. And how long do the spores stay in, like, say, stools or just the environment?
So far, we understand that these spores are really hardy. They can stay in any surface or any samples, food, liquid, or solid, or any dry places for many months, even years. Therefore, it is really hard to control them. We need to make good hygiene. We need to understand where are they sitting around. Therefore, it is really hard to control that pathogen because they are everywhere.
Yeah.
Our total environment is contaminated with fecal material from many different sources, animals, birds, insects, everything. And they also, most of the time, they may have C. difficile in their feces.
I see. Okay. Does anyone have any more questions? All right. Thank you, Dr. Alam, for the talk. I just want to make an announcement. So it appears that the Zoom link that was sent out to some of you wasn't working properly, but this session has been recorded and will be uploaded to YouTube. So we will send out the YouTube link after the session so that those of you who have missed the first part or just entered later in the session can go watch the parts that you missed on YouTube. So we will send the link to the different listserv after the session. So please go watch the recorded session. So I apologize for the wrong links that were sent to some of you. But yeah, just letting everyone know. And thank you for joining us for MicroTalks this month. And stay tuned for next month. We will be sending out further information about that. And thank you, Ileana and Dr. Alam, for the wonderful talks.
Thank you so much for inviting me.
Thank you for the invite.
All right, bye.
Bye.